Okay, sir.
Madam, good morning.
Good morning, sir.
Madam, good morning. Am I audible?
Yes, sir. Yes, sir.
Morning, sir.
Sir.
Namaste, sir.
Sir, hearty welcome, sir. Principal, sir, Dr.
Hearty welcome
Satyanarayana,
to
sir. Please welcome, sir.
sir.
Don't you? गुड मॉर्निंग कर देते हैं। चलो गुड मॉर्निंग कर देते हैं। रेस्टोरेंट भी नहीं कराया है।
Good morning, sir. Madam, chai nahi hai raha, sir.
చూస్తున్నాను మేడం జాయిన్ అయ్యారా చూస్తున్నాను.
गुड मॉर्निंग
సార్ గుడ్ మార్నింగ్.
गुड मॉर्निंग
దాస్ సార్ గుడ్ మార్నింగ్. ప్రిన్సిపల్ సార్
प्रिंसिपल गाరు गुड मॉर्निंग.
గుడ్ మార్నింగ్ సార్. గుడ్ మార్నింగ్ వెల్కమ్.
सर गुड मॉर्निंग అన్న.
గుడ్ మార్నింగ్ అన్నగారు వెల్కమ్.
Fan sound వస్తుంది.
మేడం చూడండి, మాధురి మేడం జాయిన్ అయ్యారా చూడండి.
Okay, sir.
కీనోట్ స్పీకర్ మేడం జాయిన్ అయ్యారా ఒక్కసారి చూడండి మేడం.
No, sir.
మేడం జాయిన్ స్టార్ట్ చేయొచ్చు.
Okay, sir.
మీకే పంపించినట్టు నా క్లాస్ ఆన్ చేసుకోండి.
నా క్లాస్ మీ క్లాస్ తీసుకో.
నా క్లాస్ సేమ్ నేను అన్నట్టు
చూపించు.
Good morning, Bhikshumay sir, Vishnu sir.
మేడం గుడ్ మార్నింగ్.
Bhikshumay sir, mute చేసుకోండి కొంచెం.
ఓకే ఓకే మేడం.
ఏం చేస్తున్నా తిరుపతి?
మేడం జాయిన్ అయ్యారా? శ్రీవల్లి మేడం గారు.
Sir.
మాధురి మేడం జాయిన్ అయ్యారా?
No, sir.
आप
చూడండి.
पासवर्ड ये उसको उसी से कीजिए तो मीटिंग आईडी
సార్ కాల్ చేస్తా ఒకసారి.
చేశాను, జాయిన్ అవుతున్నా అన్నారు. Joining us. Okay.
Yeah.
Sir, JDC Gambirapet, please mute yourself.
Okay.
Madam, sir, Madhuri madam has joined.
Namaste, madam. Namaste, madam.
Namaste, madam. How are you?
Madam. I'm just sharing the screen. I'm trying to.
No. First madam. So, madam, start చేయండి madam. Srivali madam, start చేయండి.
Okay, sir. Is my voice audible, sir?
Yes, your voice is audible, madam. Please.
Okay, sir. Let us. A very good morning to everyone with great pleasure. Success is not the key to happiness. Happiness is the key to success. If you love what you are doing, you will be successful. I am Srimati B. Srivali, Professor of Botany, Government Degree College, Gambirapet. I take this opportunity to welcome principals, respected resource persons, all dignitaries, research scholars, assistant professors, lecturers, teachers, students of various colleges and various departments, participants from various colleges and various states of India to this webinar. Today we are conducting a webinar in association with Government Degree College for Women, JAGTIAL, on recent trends in molecular biology by Professor Madhuri Garu. This program is aimed to give some basic and extensive information regarding recent trends in molecular biology. It is my pleasure to welcome Dr. Y. Satyanarayana Garu, Principal, Government Degree College for Women, JAGTIAL, and Sri P. Das Garu, our beloved Principal, Government Degree College, Gambirapet. Now, I wholeheartedly welcome our resource person, eminent keynote speaker, Dr. VAJHA Madhuri Garu. We welcome you, Madam Ji. I sincerely welcome Dr. Masroor Sultana Garu, Vice Principal, Government Degree College, Women, JAGTIAL. I heartfully welcome Dr. Harjot Kaur Garu, IPEC Coordinator, JDC Women, JAGTIAL, and I heartfully welcome Dr. B. Bhikshamaya Garu, IPEC Coordinator, Government Degree College, Gambirapet. I also welcome with great pleasure Sri G. Chandraya Garu, Assistant Professor, Head Department of Botany, Government Degree College for Women, JAGTIAL, and Sri A. Shankaraya Garu, Assistant Professor and Academic Coordinator, GDC Women, JAGTIAL. Our senior faculty, Sri P. Krishnamurthy Garu, Assistant Professor of English, Government Degree College, Gambirapet. Last but not least, our organizer, I wholeheartedly welcome Dr. Padala THIRUPATHI Garu, Assistant Professor and Organizing Secretary of this program. I extend my warm welcome on behalf of Department of Botany, GDC Gambirapet, and Department of Botany, JAGTIAL. Now I request Dr. Satyanarayana Garu, Principal, Government Degree College, Women, JAGTIAL. He who made his mark in the field of administration by his sincerity, regularity, dedication, and devotion in his duties. I congratulate you, sir, for achieving NAC V++ grade. We all are inspired by your teamwork, sir. Please, sir, convey your message regarding this program. Sir, please, Satyanarayana Garu.
Thank you, madam.
Thank you, sir. Please, sir.
Heartily welcome to all the dignitaries of the today's seminar on recent trends in molecular biology on behalf of GDC Women JAGTIAL, reaccreditation by NAC with B++ in association with GDC Gambiropate. We begin with, I congratulate all the organizing committee, conveners of both colleges, Chandre Garu and Srivalli Garu, and organizing secretary, Padala Tirupati Garu, and co-conveners also of the webinar from both the colleges. Convey my warm wishes. Welcome to our keynote speaker to today program, Dr. VAJHA Madhuri, Assistant Professor of Krishna University, Machilipatnam, Andhra Pradesh, and beloved Principal of GDC Women, GDC Gambiropate, Pitla Das Garu, and other aspirants. Molecular biology, it deal with the function of microcalculus like proteins and nucleic acids, which are essential to life. There have been a number of major advances in molecular biology in the past few years. So we selected this topic for the advantage to the society. Thank you, sir.
Thank you for your cooperation. Thank you very much. Now, I request our young and dynamic principal, he who puts his soul and heart day and night for developing our college. Sir, please speak few words about this program. Please, Das sir.
Thank you, madam. Good morning to all. On behalf of GDC Gambiropate and GDC Women JAGTIAL, I welcome you all. First of all, I congratulate the Department of Botany of both colleges for arranging this national level webinar. I am very much glad to be part of this webinar. I am very thankful to our keynote speaker, Dr. VAJHA Madhuri Garu, Assistant Professor, Krishna University, Machilipatnam, for accepting to be today's speaker. I think today's topic, recent trends in molecular biology, in very much useful and relevant for all the science students, faculties, and scholars. Molecular biology can be accepted to be a mainstay in the manufacture of chemicals, energies, medical, consumer products, agriculture, and food industry. I convey my best wishes to organizing secretaries and conveners and co-conveners. Thank you.
Thank you, sir. Thank you for your support and encouragement. Now I request Vice Principal Dr. Masroor Sultana a few lines about this program. Please, madam.
Thank you, madam. Thank you very much.
Thank you, sir, madam.
Okay. 1 minute, ma'am. 1 minute. Okay. Respected principal of our college, Dr. Y. Satyanarayana, sir, and principal of GDC Gambiropate, Sri P. Das, sir, convener Sri G. Chadra, sir, from GDC Women's JAGTIAL, and Srimati B. Srivalli, ma'am, from Gambiropate, co-convener Sri A. Shankaraya, sir, organizing secretary Dr. Padala Tirupathi, sir, keynote speaker Dr. V. Madhuri, ma'am, assistant professor, Krishna University, Machilipatnam, Andhra Pradesh. Participants and my dear students, good morning, everybody, and welcome to this national webinar on recent trends in molecular biology, organized by the Department of Botany and NSS of GDC Women's JAGTIAL with the Association of GDC Gambiropate of Telangana State. The study of chemical and physical structure of biological macromolecules is known as molecular biology. I think these type of webinars are very useful to our students. They mostly belong to rural areas. though they are very talented, they lack knowledge of research work. With proper guidance and motivation, they can definitely achieve their goals. So such webinars create interest in subject. Hence, and I appreciate and congratulate all the faculty members who are involved in this program. And I wish for the success of the webinar. Thank you very much, ma'am.
Thank you, madam. Thank you very much. Thank you for supporting and encouraging us. Now I request Dr. Harjot Kaur Garu, Accuracy Coordinator, GDC Women JAGTIAL. Madam, please, few words regarding this webinar.
I hope Madam is not joined. Madam will join later.
Okay, sir.
Madam, please proceed.
Now I request Dr. P. Bhikshamaya Garu, IPNC Coordinator, GDC Gambhirapet. Are you there, Bhikshamaya, sir?
Okay, madam. Okay.
No, sir. Sorry, been busy with NAC work. Now I request our senior faculty, P. Krishnamurthy Garu, Assistant Professor of English. Sir, please, few words for this webinar.
Good morning, professors. Good morning, organizing secretaries. Good morning, principals of both colleges. Good morning, the participants. It's a great opportunity not only for the students but also students, the faculty members to have very enlightened thought about this molecular biology. I will be talking about biology and get exposed to biology everywhere in the classes and outside of the classes. But they have to be given knowledge regarding what are the recent trends. What are the recent trends in the molecular biology? And really, I appreciate the hundreds of the good colleges who venture themselves to come outside and take these students to the recent trends in the vibrating of the subject like this. And this is the thing that is required by the Commissioner of Education. Not only teaching and limiting the students to the classroom, but also allowing them to know something about the recent trends. Really, it's a great opportunity for the students, really, those who are getting admitted into this webinar. So this kind of seminars are going to enlighten everyone. I really thank you very much for this opportunity. I also now, though I belong to languages, but I really feel very happy to know something about the molecular biology. What kind of the wonders that are being created in the living organism, maybe in the humans, maybe in the animals, maybe in the plants also. This is something extraordinary. I appreciate, and I appreciate our civil medical, I appreciate our THIRUPATHI Garu for initiating this kind of program. This kind of programs are much needed in the present scenario. Thank you very much for having this one. Let us tell you again.
Thank you, Krishnamurthy Garu. Thank you, sir. Now I request Sri G. Chandraya Garu, Head Department of Botany. Please, sir, few words. Chandraya Garu. Are you there, sir? Chandraya, sir?
Okay, madam, sir will join later. Okay. Please, you proceed.
Okay, sir. Okay. Now I request A. Shankaraya Garu, co-convener and academic coordinator of Government Degree College Women for JAGTIAL. Sir, please share your message.
1 second, madam. Sir is very busy, I hope.
Okay,
Madam,
okay, sir. Okay, now I request Dr. Padala Tirupathi Garu. Please introduce our esteemed keynote speaker, Dr. VAJHA Madhuri Garu. Over to Tirupathi sir. Sir, please continue the session.
Thank you, Srivalli Madam Garu. So,
thank you, sir.
We are wholeheartedly welcoming all the dignitaries and delegates of our country and respected principal sir and vice principal madam, IKC coordinator and academic coordinator, and also the principal GDC Gambhirapet, our beloved brother, Dr. Pitla Das Garu, and IKC coordinator, Dr. P. Bhikshamaya Garu, and senior faculty of GDC Gambhirapet, wholeheartedly welcome you, sir. Now, myself, Dr. Padala Tirupati, my immense pleasure to introduce one of the eminent professor, keynote speaker, Madhuri VAJHA garu, of Biosciences and Biotechnology, Krishna University, one of the esteemed university, Machilipatnam, Andhra Pradesh. Madam has a huge experience of her professional career, more than 21 years of PG teaching, and even though along with PG and UG teaching 5 years, Madam has 5 years experience. And at the same time, industrial experience. Madam has industrial experience also more than 2 years. Madam is worked as a junior scientist, Leela Nutritionscials, Vijayawada, and Madam academic career, Head Department of Botany, Andhra Loyola College, Vijayawada, Head Department of Biotechnology, Andhra Loyola College, Vijayawada, teaching as Acharya Nagarjuna University, Guntur. Now, presently, Madam has working as an assistant professor, Krishna University, Machilipatnam, Andhra Pradesh. Madam academic profile. Madam completed her UG degree in 1899. Madam has completed her PG, topper of the Central University, 1991. And Madam completed through University of Delhi in plant tissue culture biotechnology in 1993. The title is Shoot Apical Meristem Culture in Capsicum Annum. And Madam has completed her in medicinal plant research biotechnology, Acharya Nagarjuna University, Guntur. Title Comparative Molecular and Bioactive Studies on Selected Species of Calma, RBR, and Borosia, WIT, and ARN 2014. This is about Madam academic career. And Madam has also published a number of papers and published a book chapter. A book chapter title is Evaluation of Depigmentation Activity of Xylea, Xylocarpa, and its phytoconstituents is very well written and has been accepted for publication in edited book title Advances in Biotechnology and Biosciences, Volume 6 in 2020. Madam has conducted a number of conferences, webinars, and also Madam published a number of reputed national and international journals. Madam has this huge experience is very useful for today's webinar, national webinar on recent trends in molecular biology. This is also very useful for all the UG students and also PG students of the biotechnology and the life science stream, and also it's helpful for all the faculty members, those who are working in the various degree colleges and PG centers of the various universities of our nation. So we are wholeheartedly welcoming you today's keynote speaker, Professor Madhuri VAJHA Madam Garu. We are wholeheartedly welcoming you, Madam. Please start the session. Madam, please unmute. Madam, please unmute yourself.
Yes. Respected dignitaries, whoever present here, respected Principal of the GDCW, JAGTIAL, Dr. Y. Satyanarayana Garu, respected Sri Pitla Das Garu, Principal of GDC, Gambhirpet, and respected conveners of today's national seminar, Sri D. Chandraya Garu, B. Srivalli Madam, A. Sankaraya Sir, and also today's organizing secretary of today's one-day national seminar, Dr. Padala Tirupathi Garu, and other faculty members and the vice principal of college, Manasur Sultana Madam, and other faculty members, my dear students, research scholars, very good morning to one and all. So today I am going to talk about, I'm going to share now the screen, please see all of you. So today I am going to talk about crop improvement, how the crop improvement can be taken place. So crop improvement is very much important for everybody because with respect to the increasing the crop yield, nutritional quality, and if you want to grow them in branching, high branching, dwarfism, and maturity, and the duration level reducing, pest resistance, disease resistant, whatever may be, all these things come under crop improvement characters. This crop improvement can be achieved by simple methods or by plant breeding methods like mass selection, backcross selection, self-pollinated, cross-pollinated, and also vegetative propagated plants. There are several methods are there in plant breeding. Next level comes to the genetic engineering method, where you can do the crops genetically modified in such a way you can improve the crop. So genetically modifying means you are creating a plant with desirable characters. Obviously, you concentrate on the improvement of a crop. The next comes to the after transgenics marker assisted selection. So you can select the plant with a particular marker, molecular markers like RFLP, RFPD, all those things, and you can select those plants also. And the next now, it recently developing area is RNA interference. This is also one of the mechanism, one of the method that is applied for the crop improvement. So what is the role? What is the mechanism? How the applications of RNA interference you can observe while doing crop improvement. So not only this crop improvement helping the plant to be improved, and also one can earn money also. So that you can call it as biotech's $1 billion breakthrough. So this RNAi for the last decade, it is growing a lot. This is a growing field in the molecular biology. So stepwise, we will go one after the other. So here, as we know, the production rate of the plants is getting decreased, the population rate is increasing, rapid urbanization, industrial belt, all these things, soil erosion is taking place, climate getting changed, pollution is taking place. So natural resources are getting depleted, land, water bodies are getting depleted, and you need to improve the seed, you need to improve, or you need to develop disease resistant and pest resistant plants. And also at the same time, the plants must contain effective plant nutrients. They must be resistant to abiotic stress. They must be resistant to environmental problems, whatever occur day to day. So you need to go for sustainable technology, the technology which is useful to maintain the plants yesterday, today, and also tomorrow. So you have to invent for that and concentrate more on that subject so that you can get sustainable rate in the crop improvement. So why we should go for the first thing, simple farming. The second level, plant breeding. A third thing that comes is transgenics, genetically modified crops. And the next thing, RNAi. So why we should go for GM crops, genetically modified crops, genetically modified foods? What is the need for that? So there is a challenge for that. The population is growing day by day. Human hunger diseases, malnutrition is high level in this. So there is a global demand is there for increasing the food products, for improving the quality of the crop, for improving the food production. So the technology options are plant breeding, then genetic engineering. So there is a need for second green revolution. So then what you see here, there are agrobiotechnology, biotechnology. There are two terms are there. Biotechnology means biology field. So there is a basic needs for food, clothing, shelter, health, safety. All these must be improved. In agrobiotechnology means in agriculture, in crop fields, you need to improve the quality, you need to improve the pest resistance, disease resistance, bacterial resistance, pathogen resistance, abiotic stress resistant, and also all nutrition important characters. Not only that, visible characters also. For example, here, but there are some characters human desires, but they are not available in the fields. So what you have to do, you have to create, you have to go for the technology, new technology. So here the fruits normal banana fruits we take, they don't have vaccines. But if you produce the fruits with vaccines, so blue roses and also cotton, so something like that, you have to do like. So there must be a plant with a desirable character. There is a plant. What is this plant? This is a banana. A desirable character is having vaccine. Simple, by eating banana, you can get vaccinated. And also similarly, it is rare to get blue roses. The blue roses, you can see pink roses, white roses, but people want to see blue roses. So there is a technology. And also blue cotton. These simple things. So technology is for this. So the grains with improved nutrition. So these are all strawberries, genetically modified strawberries. So these are all the plants which are improved with their qualities or color, or maybe showy appearance, or visibility, or nutritional quality, whatever may be, you are changing the character. So there is one industry that is name is Gentech. Recently, genetically manipulated corn they developed. So what if you eat this corn, fat is burned. So obesity will be reduced. So that's how they concentrate on one particular character. Simply by eating this corn, one can reduce their fat. So the industry that developed is Gentech industry. So organism who eats this corn, he starts losing his fat deposits. So like that, the technology is playing important role. So after plant breeding, you can see they're making a transgenic approach. Transgenic means transfer of a gene, a foreign gene, into a plant where it is not there. So you can transfer a particular gene using a binary vector, that is agrobacterium mediated tumefacts. Now all of you might be knowing about agrobacterium mediated transfer. So you are transferring a gene using a vector. So you are using suspension culture. What is suspension culture? What is inoculation? What is a vector? What is the gene gun? How do you produce all those things? Once, see here what you have to do. If a gene of interest, that means a particular gene is responsible for a character. So that gene of interest, first you have to isolate. And that gene of interest must be placed in a vector. A vector is a vehicle. A vehicle is the one which carries the genetic information from one place to the another place. So in this vector, you are tagging your gene. And through the agrobacterium you transferred it. And this agrobacterium is inoculated to the hypocotyl region. You know what is cotyledon? Cotyledons of the first leaves. Below the region you can find hypocotyl. Above the region you can find epicotyl. So at that epicotyl region, you inoculate with the agrobacterium and you place it on MSC medium. And then slowly callus is formed. And the callus, if you suspend in the liquid medium is the medium that is a morass. You can call it as suspension medium. In this suspension medium, just keep it and put it in the shaker. You will find the cells and also sometimes embryos. Embryos means those embryos, heart-shaped, torpedo-shaped, globular embryos you can find. These embryos, they will give rise directly to the shoots. The shoot induction medium is supplemented with the cytokinins, and the root induction medium is supplemented with auxins like naphthalene acetic acid, indole acetic acid, or 2,4-D, sometimes with permutations and combination with the auxins, and it may change from species to species. You can find the shoots. After developing the shoots, you have to go for the root induction medium. Thereby, you will get a plant, and this plant is called transgenic plant with a desirable gene. Previously, the plant is without that gene, but after introducing that gene, that is called transgenic approach, this plant now having the gene of our interest. So like that, the corns, like that, all those blue roses, those are all developed. So for everything, it takes so many years for doing research, for the development, for getting commercialized, that means marketing and earning money. See how many years that is taking place for the trait generation, optimization, and also you have to form a concept or a model plant, and then testing, and then field trial. whether that plant can survive in the field in the normal conditions, or only it can survive in greenhouse effects. Then finally you have to go for commercialization. Like that, it takes so many years. Now, recently, I think 2008 to 2003, something like that, RNA interference. There is another technology, there's a sustainable technology where one can improve the crop character, where one can produce disease-resistant plants, where by using, you can do virus-resistant plants, worm-resistant plants, where you can destroy the viruses, you can destroy the bacteria which will attack the plant. So that technology, that sustainable technology is now called as RNA interference. RNAi. I means interference. You know, all of you know what is the central dogma. So gene is a segment of DNA. The DNA is the double-stranded DNA, out of which one is the target DNA. From the target DNA, you will get hnRNA. So how much ever length of DNA is there, you can find the similar length of hnRNA. hnRNA means heterogeneous nuclear RNA. It consists of exons and also introns. The introns are cleaved off and the processing takes place. Thereby all exons make up mRNA, that is messenger RNA. The sequence of nucleotides, ribonucleotides, determine the sequence of amino acids in polypeptide. Thereby you will get a protein. So what is the central dogma? DNA to mRNA by transcription, mRNA to polypeptide chain by translation is a central dogma. In this central dogma, these polypeptides make a protein, and all enzymes are proteins. The proteins include structural proteins as well as functional proteins, which will create the phenotype. From the phenotype, you can say this particular protein is there. To go back, that particular DNA is there. So if you silence that particular DNA, mRNA is silenced. If mRNA is silenced, no more protein. If no more protein, that particular phenotype cannot be seen. This type of silencing is called gene silencing. So what you are doing, you are suppressing the gene, not to go for mRNA. If it is not, mRNA is not formed, that polypeptide is not formed, so that phenotype is stopped. That is called gene silencing. It is of two types. Gene silencing you can do at a transcription level. Gene silencing you can do at a translation level. Transcription, as you know, from DNA to mRNA. Translation is mRNA to polypeptide. So here, these are the basic things that you should be in our mind before understanding the technology. The technology of RNA interference, how it is useful, how you can do research, even you can do, if you learn the technique, you can do miracles in the lab and you can produce whatever you want in plants. Only thing is you have to practice the technique and the basic knowledge you have to know. So here today I'm going to deal with that RNA interference. As you know, RNA means mRNA. mRNA means messenger RNA. RNA is always a single stranded. RNA consists of ribonucleotide sequence. Okay, so this RNA, how it is RNA interference.
Excuse me, madam. Please, excuse me, madam. Sorry for the interference. Sorry for the disturbance, madam. So, this session will going to wind up this session. Please, all of you, all the delegates, personal requesting you, please join through same link.
Same link, okay.
Madam also, madam, all of you join through same link after wind up this session.
Okay. Okay, sir. Thank you.